Okay, sorry, my voice has just decided to break at the wrong moment. So good afternoon, everybody. My name's Chris Mann. I'm from this company here called Asphalion in Barcelona. So I'm going to give a talk that's quite different from all the other ones that we've had this afternoon, because I'm coming from a regulatory point of view, not from a data clinical point of view. But actually, this will be a very good summary, I think, of quite a lot of the concepts that we've seen today. And a lot of the words and things that have been said will come up in a different context. So let's jump in, because we've only got a bit of time, and I think Christian said there was a few beers up there, so he knows how much I like that. So, all right. So I'm going to talk to you today, as I said, about commercialization of medicinal products, and I'm going to focus on two types very quickly: repurposed drugs and gene therapy. Okay. So first, just a bit of background. I mentioned I'm not doing research, or I'm not a clinician, but I used to be a researcher. So I have a background in gene therapy, and I was in the lab for about 15 years. And I thought, well, these guys don't like their photos, but I put some up here, which I love. So this is an old one, actually, from 2002 when I was writing my PhD. You can see my old computer there, and all this is full of my papers. But my PhD basically became this product here, which is from Sarepta for treating muscular dystrophy. So I'll talk a little bit about muscular dystrophy as an example, okay? But basically, since then, I left the lab in 2012, at the end of 2012, and I've been doing regulatory since then. So my expertise is gene therapy, biologics, and actually pediatric medicine that works really well. Okay, so I'm going to talk to you about a couple of things today. This is just a quick overview of the structure. So I'm going to give you an overview of drug regulation. I'm going to highlight 3 pillars of drug development that we'll come back to here and try and apply each one of those to advanced therapies and medicinal products. But before that, I'll give you a little bit of a definition of what these really are. And unfortunately, I have to talk a little bit about some legal stuff, because that's what I do. And you can't kind of get out of it, basically, for this. And then we'll also talk about a few other regulatory concepts on the way, okay? But firstly, a quick disclaimer. Obviously, if anyone's ever worked with anyone like me, regulatory, we talk in these weird codes all the time. Like, we always have these numbers, names, letters, and we know what we're talking about, but nobody else does. But I'll try to keep it minimal today as much as possible. But the key point here is this is highly specific language. Sometimes it's totally controlled with no flexibility, and it has legal implications. So if I say something wrong, someone will sue me. So that's always important. Okay, so let's start right at the beginning then, so we can tell as simple a story as possible, and it will be exaggeratedly simple, so hopefully you can make some questions. But basically, pharma industry, as you know, it's a regulated industry. That means there's rules and regulations and laws that describe what you can do, so you don't end up with this kind of situation. Stem cell clinics and stuff like that have been around for ages. When I did muscular dystrophy, for example, in the end of the 90s, there was some giving myoblast transfer treatments, which were clearly scandalous and not doing anything, but they made some money out of it at the time. But here are the 3 pillars, basically. So the regulations set up this requirement that you have to show quality, safety, and efficacy. So quality basically just means you can manufacture your product to a good consistency and standard. Safety means there's no risk to the target population, and obviously efficacy means it actually does something, what you say it does. I'll go into details on that in a minute. Okay, so in the EU, at least, the commercialization process is based around the marketing authorization application. So I'll call it MAA sometimes. In the FDA, it's called something different. So the MAA includes all your summary of all of this stuff, and obviously the legislation defines all that. But obviously, you have to be a bit careful about the regulation and what's going to change, because this is something very new from our friend Robert Kennedy, who's basically going to dissolve FDA next week or in January when he gets to be head of health in the USA. So keep your eyes on that one, okay? So jumping back to the 3 pillars then, let's just go through it a little bit more detail. So we've already talked about the compliance of this is necessary. So quality, what we really mean by quality is chemistry, manufacturing and controls, CMC we call it, or it's basically manufacturing. So obviously they want you to build quality into your manufacturing process, so you're not making crap, basically. So the key point here as well is your manufacturing process, or your CMC, really defines your product. There's a classic cliche in regulatory, which is your process equals your product. And I'll explain a little bit how that works in terms of repurposed drugs, especially. Okay. The key thing as well, quality is throughout the whole development of your product, not just at the beginning or the end. Okay. So when we talk about safety as well, we're not actually really talking just about safety, but we're really talking about non-clinical or animal studies. So showing safety profile before you get to clinical studies. So the safety or non-clinical studies are usually at the beginning of the development before human exposure, and includes things like genetically modified organism or environmental safety, as well as patient safety. Safety, I would just add as well, it's always tested in human trials. So here we're just talking about non-clinical studies. It's just another way of saying the same. And efficacy is the same. It just means clinical studies, basically. And some of these may go beyond commercialization, okay? So this is a really simple overview of a classic medicinal product. So we'll show you some variations on this later. So basically, this is what you get. You start out in the lab with your hypothesis, or in this case, we've been talking about models and things. You have a kind of mechanistic studies, then you do dosing studies, then you do toxicology. That's your safety assessment there in green. Then you go into the clinic, so you have a classic phase 1, phase 2, phase 3. I think these guys have just talked about a phase 1. We'll show you a little bit about that later, but that's efficacy. But obviously, as I said, safety is in there as well. Manufacturing, as you can see, goes all the way through. It starts not right at the beginning, but it starts when you start doing regulatory preclinical studies, and goes all the way through post-marketing and stuff. The key point is this: your marketing authorization application. This is submitted by a marketing authorization holder, so a company, an individual, but usually a company. They submit the dossier, there's a benefit risk assessment, and if it's okay, you get the summary of product characteristics, which is the label of the product that says how it should be used and why it's used in that way. So obviously we've talked a bit about this. Off-label use is if you have a use for the same product that's not in the actual SMPC, okay? So that's really basic overview. So we'll go through some of the concepts. So very briefly, right, we need to talk about this new active substance. So this is basically just a new drug, something that's never ever been used before in a medicinal product. So when we talk about drug repurposing, sometimes it's called repositioning. We're talking about not new active substance, but old ones, let's say, that are just used in another way. So sometimes people talk about repurposing, which is maybe you're already on the market and you just convert it with one of these kind of applications into a new product. The other way is if you take a product that was evaluated or never got to market, but it's no longer commercialized, and you reposition it to be on the market, okay? Usually this is linked to patents, and if they're off patent and this and that, but that doesn't always have to be the case. But basically the key point is repurposed drugs or new drugs will still go through the same approval process. So the 3 pillars apply to all of them, okay? But we'll show you where that varies a little bit. So looking at the types of active substances then, what are we talking about here? So these are really all different types of drugs that you can have. So small molecules, we've seen a few of those, antibodies, you know that. Obviously the COVID vaccines, which are going to get banned under Robert Kennedy in a few months' time, so no problem about that. And then you have all these more complicated biologics products here. The key thing here is all of these advanced therapies, ATMPs sometimes called, and you have a subset here in green, which are the gene therapies, so viral vectors, gene editing and stuff like that, okay? There's also this class here at the top called medical devices, which are not drugs per se, but they overlap. You might have nanoparticles in some of these things, but some of these techniques, concepts may also link to your medicinal product. And I'll show you the example of the companion diagnostic later, okay? So this is the most boring slide. So if you're ready to sleep, this is the best one to have a sleep in. I'm not going to explain all the details, but as I said, I have to put this legally here. But and this is the only one where I really talk about the US stuff as well. But basically, if I want to make a medicinal product and commercialize it in the EU, there's 2 steps. First one is the legal basis. What is the framework legally that I have to operate? So this really depends on what type of product I have. For example, the new active substance substances always under this basis. Repurposed drug may be this kind of mixed dossier, but sometimes you might also have fixed dose combinations, so two substances. So basically any of this more or less applies except for the generic hybrid ones. In the US, it's a little bit simpler. If I was a gene therapy, I would be sorry, not a biosimilar. I think I missed one here, sorry, that's a biosimilar. But basically, if I was a new substance, I would be here, B1. And if I'm maybe a repurposed drug, I'll be B2. So that's kind of a boring thing, but it's really critical to have this definition, and it really depends on the product that you're talking about. The other thing that gets confusing in the EU, and some people have made some comments about this, is also this point, the review procedure, because we have more than one country, so I can send my dossier to one country at a time, two at a time. I can send it to all of them except for Sweden if I want to, using these procedures, and they're reviewed by the national agencies. But obviously the EMA or the European Medicines Agency is involved, and they do what's called the centralized procedure for some products. So I highlight here, it's mandatory to go to the EMA for neurodegenerative diseases, advanced therapies, and also orphan medicinal products. So basically we're talking about EMA for that. And I think this is just a summary of those points, but maybe this is the one just to emphasize. So the legal basis defines the overall data requirements and where you go to review it. That's really the main thing from that, okay? So if I apply then these three pillars, then we'll start going through a theoretical example. It doesn't really matter what the drug is. We're just going to go through the main stuff, okay? So what you basically have is a repurposed drug is one that already exists. So what you would have usually, we've talked about the marketing authorization holder before. They might already be making what we call the drug substance. That's not Charlie Sheen's breakfast. That's just this purified active substance, basically as a way that with pre-formulation. So obviously I can make many different types of pills, capsules, injections from that, and it's still the same substance, but it's just a different formulation. So depending on what they've got on the market, you might just get this from them, or you might get this and start doing studies as well. If the drug was kind of off the market, you might have the manufacturer who used to make this and they still have the process. So you can basically come along and say, can you make me that? And they'll say yes. And they already have all of the qualifications, specifications and stuff like that. So it's pretty easy to do, basically. Okay. But the other thing that comes up here with the formulations is this. Do you need to adapt that formulation for the repurpose use? You might have a new route of administration. That might also imply a patent. And I've just put this here, and I'll come back a little bit to that later, that pediatric patients need different formulations. They can't swallow pills. And you guys were talking about the feeding tubes, for example. That's a really important element here. You know, the compatibility with the tubes, can you use it in a tube and so on. So they're really key elements to think about. But when you get to advanced therapies, you have a completely different story. I've just put a theoretical manufacturing process here for AAV, or adeno-associated viral vector. This is what's called triple transfection. It doesn't matter what it is. It's just there to show you it's complicated. You have bacterial cells, human cells, big bioreactors, purifications, and then you get this kind of virus at the end. But basically, because you're talking about a new active substance, basically you have to set up a new manufacturer and find who they are. Then you're linked to them. As we said, the process is the product. And sorry, that goes back to the previous slide as well, where you're buying the product definition by the manufacturing relationship, basically. So now with AAVs and HMPs, there's a lot of experience, at least up to phase 1 and 2, but sometimes commercial manufacturers aren't really around. And obviously setting up all this, maintaining this, it has a lot of costs. So the difference between a repurposed drug ATMP really comes down to the manufacturing process in terms of cost. Okay. The other thing that comes up with rare disease is if you have to do all this, you make a big batch and you get 2,000 vials, that might be 1,800 more vials than you have patients. So suddenly you just have a big freezer full of this, and that means you can't do multiple batches because you don't need to do that, etc. So that has regulatory implications at the end. And I've just put this comment in green. This is a little bit hearsay, but there is rumors that there's going to be a change in the future about orphan drugs, that there will be more flexibility flexibility around manufacturing, especially for advanced therapies, where you can kind of leave them, let's say, in a phase 1-2 environment because of all this situation. Why do we have to keep manufacturing just to make you happy when we're not going to use that? And it's expensive, and obviously getting the cost down is something else. Okay. All right, so here's a quick summary of safety. Okay, so I've put this basically in a table at the bottom. So you remember the green things on the slide before. We had our PD, PK, and tox type studies, okay? So this is mechanistic studies. So if I'm repurposing a drug, I still probably have to do some of that for two things. I have to show proof of concept, and you guys have really highlighted that a lot. Mutation A doesn't mean mutation B, so maybe I have to show a mechanism in all these different mutations to then bring them into the clinical trial because I have a proof of concept. If I only study one mutation type, maybe I can't bring the others into the trial, and that's the same for ATMPs. And obviously the key is the dose determination against those receptors or different things as well. In terms of dose levels, pharmacokinetics, basically you might not need to do anything for repurposed drug. It really depends if you change the route of administration, the formulation, maybe the dose level. If you go more than what it was approved before, then maybe you have to do something, and that goes for tox as well, if the same thing. But for an ATMP, because you're a new active substance, you have to do everything. And basically you have a few concepts that are a bit different. So biodistribution and shedding is not the same as PK. And I've shown you an example here of biodistribution just to highlight a really key point. And I'll come back to that picture in a minute. But you also have other things like integration from these vectors, immunogenicity assessment. And I'll show you the example of that relation to the CDX in a minute as well. But you guys have talked a lot about models and things like that. So there's a lot of questions here for me about this level, much more than you'd normally get or expect for choosing the species. Is mice the best one? Is that model the best one? How many models do I have to do? And all that kind of stuff become key questions. But just to return to this kind of highlights that choice as well and how much work you have to do. So this is a biodistribution study for I think it was systemic injection of a vector. So this is AAV9, which is serotype 9. So basically, if I do IV, it doesn't go to the brain. But these guys made a special vector based on this called PHPB that was selected in C57, and it goes really well to the brain from intravenous administration. But these guys found out by coincidence it doesn't work in bulb C, okay? So that just gives you an idea of how important some of these studies are, and just to be careful about what's going on, because even with the animal models, you can get a lot of difference, okay? And this slide here also just emphasizes this, but on a different way, okay? So here I'm talking about orphan drugs, so rare disease drug requirements. So here is the EU definition. I think someone had a previous slide which showed this. So basically, to get an orphan drug designation in Europe, you need three things. You have to have a life-threatening or critically debilitating condition, which GRIN disorders are. Prevalence has to be very low. So you can see here the classic curve. So we're talking about very rare diseases down here with very few patients. So that's usually the case, but you have to show a good prevalence as well. And then just two quick concepts. So there should be no treatments, or if you have a treatment that exists, you have to show a benefit against that. So with GRIN, I think we talked about Christian about this. What are the treatments in what type of patients, in what moment, etc., is not clear. So having to show that or not is unclear. And sometimes you may need to show that in an animal model before you apply for your orphan drug to be able to get the orphan drug. So therefore you need to know what the treatments are and if they work in your animal model. That's another thing. But I wanted to highlight this concept just a little bit more as well, medical plausibility. This is much more complicated than it seems, because you might have a gene therapy vector, you express the protein, and that's pretty easy, but it's actually not. And I've given you the example here of muscular dystrophy again. Okay, so this is three animal models which are described in the literature here. So the classic MDX mouse, which basically doesn't reproduce the pathology of human patients. The mice have a normal lifespan, not really much muscle damage, etc. Cardiophenotype's quite weak as well. So they made a double knockout mouse that may be a bit better, but actually that doesn't reflect the human disease because it has two genes missing and all these other things. Then there's another one. This has these calcium input accumulations, and then you think, well, which one of these is the best one? And here's just an example of gene therapy, and this is a really key point. So this is what normal muscle looks like. You have dystrophin at the sarcolemma. The MDX mouse, which is a knockout, has none, except for one fiber here, which is called a revertant fiber. I'm not going to go into that now, just forget about it for the moment. And here's gene therapy. So you restore dystrophin to where it was missing. So that looks good. But the point here is this is not sufficient to get your orphan drug designation. And the reason for that is this is a biomarker, and we've talked a bit about this later, and I'll show you another example. It doesn't mean anything. These mice have no pathological features. So if I add this, I'm not improving anything. So therefore, I can't show that I'm going to have a benefit in the clinical population. So there's many, many examples like this that come up from rare diseases and these animal models. So you have to be careful. So the question here is really, how many fibers do I have to get dystrophin in, and how much dystrophin do I need in that fiber to then say this will have a physical benefit to the muscle? Okay. Okay, so clinical, I kind of have to skip a bit because of time, but I've just made one slide just to show you what clinic does look like for rare diseases and especially for ATMPs. So if you remember before, we had phase 1, 2, 3 and all that kind of stuff. But here, basically, you might have a completely different clinical thing where you have a phase 1, 2 study, but you've only got 20 patients, but that's enough to go to marketing already, or at least a conditional marketing, because you've compared to some kind of real world study, natural history study, which we've also heard about from here somewhere. And I've put this starting before this study, because maybe you need some data to then decide on the design of this or to make sure the endpoint's the same, and obviously has a long period of evaluation and so on. The other thing for gene therapies, especially it comes up, is long-term follow-up or LTFU. You have to do 5 or even 15 years, depending on the vector, following up the patients here have been treated in case you have integration and stuff like that. So that's a thing to keep in mind. And I've also exaggerated and made this conditional marketing authorization, which happens when you go to marketing with a biomarker, for example, and you need to wait for some of this data, or in exceptional circumstances where you really have so few patients, you'll never, ever get good data. So obviously, assuming that all works well, then you get your marketing at the end formally. Okay. So this is the classic biomarker thing that I wanted to mention to you. So I think it's easy to explain just up here. So this is basically Sanfilippo A syndrome, so mucopolysaccharidosis type 3A, which you've probably heard about. It's obviously neurodegenerative as well. So this figure just shows you the patient age in time with different functions that get lost over time because it's a progressive disease. So basically the point is, if I do a clinical trial starting at 4 till 6, some of these functions will not change much in that time, so I can't show clinical benefit against those symptoms, right? So the idea then is I develop a biomarker which shows that I'm having progress because I can relate this biomarker to a functional outcome later. This is data from actually dogs from my old lab, not my paper, but my old boss did this paper. But basically the point of this paper is saying, if I inject gene therapy here at day 0 in these dogs, I think this was probably liver gene therapy, what you get is systemic sulfamidase activity, which you can measure, you know, like an ELISA. And you see a peak here, it goes down a bit because you probably get an immune reaction, and then it's more or less stable, but slowly dropping off. The key point here is this is a direct result of gene therapy because you have 0 if you don't. If you can correlate this with activity, which you can in this disease, you know that a level of at least 2, I'll invent, you know, it gives you efficacy. So if I can show that this is above 2 for a period of time, then I'm going to show that I'm going to have some benefit probably with time. So that's really the classic biomarker approach for gene therapy. So I would go to marketing here rather than waiting 5 years for each of the patients, okay? I just wanted to talk about this very, very briefly, or just mention the concept, okay? So there's something else here as well. So qualification of clinical endpoints, including biomarkers, is really fundamental. And I've put the opinion here. This is from the EMA and a specific procedure for muscular dystrophy again. So you can see it's actually just a walking test. So basically, they've just validated and said, this test, if you do it in this way, is a clinical evaluation of these patients. But rather than just say, we did this and it is, you have to accept it by the community, by the regulators. So you go to the EMA, you propose the endpoint and say, we want to qualify this, and they give you the outcome in a procedure. That's a simplified version of that. And then just to highlight this is a CDX. So this is for a real product. This is approved by FDA, but basically, I can't remember which one it is, one of the hemophilia gene therapies maybe, but basically you cannot treat all the patients. You have to screen them for immunogenicity. If you have antibodies over a certain level, it's a safety concern, so you're out of the trial. But the test to do that screening is forcing, let's say, a clinical decision, which means you have to develop what's called a CDX to go with your product to then, to be able to make that decision consistently. So that's another thing to keep in mind while developing this, okay? And I think this is the last slide, just to go through this important element, especially for this kind of condition, okay? So just to emphasize, in Europe, and this is more or less the same in the US, you have a pediatric regulation that basically says you have to be compliant with this at the time of marketing, or you cannot go forward and you cannot be commercialized. So basically what it says is you need pediatric compliance here, you need a pediatric investigation plan, which includes pediatric study data, includes a waiver for doing those studies because the disease doesn't exist or you can't do it or whatever, or a deferral because maybe you need safety data from adults or something and then you go. But this plan has to cover from birth to less than 18, so the whole pediatric age subset, okay? So when you do complete this, you get basically extension of your patent, which may be important, especially in some applications. Gene therapy, you would have a patent, maybe in some repurposing you wouldn't. But I've also highlighted this, that if you have an off-patent drug used for pediatric medicine, you may be able to get one of these licenses that would then give you a patent, basically. Okay, so the usual plan for a pediatric relation with the EMA is you would submit the PIP after the adult phase 1, but if you've got purely pediatric, maybe you would do it early. And some of the key issues we've already discussed a little bit. Is the formulation good? Because they might oblige you to change the formulation to suitable for neonates, for example. You also might have developmental toxicity, and I think that was touched on by somebody as well, so maybe you have to go back and do tox. And also you might have to do dose adjustment studies or extrapolation from clinical stuff. And then you do your pediatric compliance, okay? So that's the end of the talk. I think I'll just leave this here to have a look at. This just summarizes the main points I've just mentioned here and what the key concepts are for a repurposed drug or an ATMP. And I just chucked a few of these things on the bottom just for context. These numbers on success rates are from the literature, but probably maybe high for an ATMP even. But basically, that's it. So you can see quite a few differences, but the overall thing, this should be cheaper, quicker, and more successful than doing a new ATMP, but it will depend on your final mechanism, okay? So this just say thanks to everyone in our team, and we also have funding from some European projects, but we're not a researcher. So thank you very much.